Oh, I'm sorry. All right, we'll just wait a couple minutes and get everyone here. So just one thing I need to open up. Way more organized than I thought it was. Okay. All right. Well, got 4 of you. Hopefully the next 2 minutes that increases by 12. We're going to finish off talking about the eye, and we're going to talk about the ear today. What all those things are about. And if you had a chance to print out the this out. If you didn't, that's fine. I'll show you how to use it to find your blind spot today. So we'll do that. I'll just give a couple more minutes for people to show up here. And then we'll get started with the last class of the week. What I'm thinking, depending on how far we get today, I'm hoping we just finish with the nervous system today. You'll have a worksheet that'll be due on at noon on Monday. And then I think Monday's class will just be a check-in class. So I'll just check in with people on Monday rather than us continuing on. And I'd like to promise you a Kahoot, but I don't think I'll have time to do that. So, but we'll just do a quick check-in class and see who's around and who has questions, because you'll have the quiz, but then I'm going to open up a unit exam for you guys next Friday for the nervous system. But on Wednesday's class, we're continuing on with reproduction, our very last unit of the year. So just wait one more minute here for people to show up. Oh, there we have Kaylee. All right. And it is 2 o'clock, and I only have 5 students. Yeesh. Okay, well, that's all right. We will continue on. Everything's being recorded. Oh, and I see something on chat. Sorry, I missed that earlier. Ooh. Yes, Eric, that is a mistake. That's not supposed to say June 1st. Sorry, I got my times wrong with that. That is supposed to say May 25th. I will go in and change that in a little bit, but it is supposed to say May 25th. So I will fix that. Thank you for letting me know. Yeah, so the worksheet that you guys just would have received at 2:50 today, or sorry, 1:50 or 1:55, I can't remember the time I put on it, is due May 25th. So I will go in and change it after our class today. So just so you're aware. So that is this coming Monday. Sorry about that. Okay, let's get started. We are going to take a look at special senses and finish that off. So I will share mine with you. So let me just go to my share here. I'll find it. And there we are. And we're just going to go all the way back to where we're that. So the last thing we talked about was aging and the effects of aging on your eyes. So we want to make sure, or you want to make sure you know all the different parts of the eye like we talked about last class. There. And to go here. We want to know that we've got two autonomic muscles, or two muscles that sort of are autonomic for us that are in our eyes. And one of them is the ciliary muscle that helps to control the shape of the lens. It actually attaches to ligaments that attach to the lens, but ultimately it's the muscle that controls the ligaments which control the lens. And then we have our iris, which remember are muscles that control the size of our pupil. Okay, so we got to this point here, so I'm just going to open that up. Aging, so we talked last class about how as we get older, we have more protein buildup on our lens, and as a result of that, our lens loses its flexibility. Okay, so you can explain to your parents what's going on with their eyes as they're trying to hold things out further and further in order for them to see it. Okay. All right. So what are the steps? We want to know the pathway through the eye, and we should understand every time the light hits a different medium, it bends. A lot of bending happens. So the first bend, light's going to come from the air and it's going to hit our cornea. Well, that's a different medium than the air, so the light bends. Then the light's going to pass through the aqueous humor, different medium, light bends. Okay. Then it's going to go through the lens. Now, it has to go through the pupil to get to the lens, but the pupil's not a different medium, the pupil's just a hole, right? Goes to the lens, it bends. Then it travels through the vitreous humor, which is that jelly-like stuff that's in the center of the eye, bends again. So those four structures, the cornea, aqueous humor, lens, and vitreous humor, are what allow that light to refract, and all of that bending results in light landing on the fovea centralis, right? That's the center vision. That's our central focus point, is the fovea centralis. Okay. Now, light's going to hit on either side of the fovea, right? So light that's sitting over here on this side of the periphery is going to be my peripheral vision over here, and over here will be my peripheral vision. As soon as I look at it, that now becomes my central vision, and it hits the fovea centralis. Okay. Photoreceptors stimulate sensory neurons, which are in the optic nerve. So I named the optic nerve. So because I named it, you need to know it. So the optic nerve, which is going to send that message to the occipital lobe of the brain. Okay, so I talked about that central vision. Fovea centralis is where our central vision hits. So if it's hitting there, what type of photoreceptors sits on the fovea centralis? What type of photoreceptors sit on the fovea centralis? There's two types of photoreceptors, so in your chat, tell me one type. The type that is on the fovea centralis. Okay, thank you very much. The cones are on the fovea centralis. What types of receptors are outside the fovea centralis? Yes, rods are on the outside, right? So theoretically, I shouldn't see color over in my peripheral vision. I do because my brain fills stuff in for me, okay? All right. So cataracts. Cataracts happens when our lens becomes opaque. So we start getting all throughout the lens rather than just the surface. The surface, when we get older, is just a transparent protein. Light can still get through it, okay? It's just the lens loses its flexibility. A cataract is where we get cloudy material within the lens. So when you look at someone with a cataract, you're not going to see clearly right into the back of the retina, okay? So that causes light to kind of disperse rather than hit nicely here on the fovea centralis. So the lens becomes opaque, preventing passage of light. And what they do is they replace the lens with a plastic lens. So we're just going to watch a little surgery here on that. So let me just find my open all these up and dig up. Okay. Oops. Okay, well, I just want to go here. And then, sorry, I'm going to go back to my share so we actually have sound. There we are. Okay, so we are going to look at just a little cataract surgery, like so.
This operation is called phacoemulsification cataract surgery. Click the navigation arrows below the animation screen to play, pause, rewind or fast forward the animation. Here we show the different parts of the eye. A cataract of your eye. It usually develops over a long period of time. Most often a cataract is due to the aging process. Once your ophthalmic surgeon has examined you, drops will be placed in your eye to dilate or widen your pupil. This makes it easier for the surgeon to see the lens of your eye. Local anesthetic drops will be placed in your eye. This will completely numb your eye. Sometimes a local anesthetic injection is used instead of drops. A tiny cut is made on the surface of your eye. The cut or opening is usually so small that no stitches are needed after the operation. An ultrasound tool is used to break up the lens.
So basically the lens just gets chopped into a zillion
The
different
lens
pieces.
sits in a thin sac of tissue called a lens capsule. The broken up pieces of lens are removed from the lens capsule through a small
A little
tube.
vacuum comes in and sucks all those bits out.
An artificial lens or intraocular lens is inserted through the cut into the lens capsule. The lens is folded so that it can pass easily through the tiny opening. Once inserted, the artificial lens unfolds itself and replaces the old lens.
Now, in the old
Your eye will
days,
be covered with a dressing to protect it.
in the old days when they did this surgery, they didn't replace the lens, they just took out the lens, which meant that people would have to wear very, very thick lenses on their glasses in order to accommodate for that. Where now, it doesn't mean you wouldn't wear glasses at all. You would now have monofocal, so you wouldn't be able to see, your lens wouldn't adjust to near and far. Although I think there are some lenses that have come out that are multifocal lenses, but I'm not totally sure on that. Okay. All right, so let's go back to the PowerPoint here. Okay, so nearsightedness and farsightedness. Now I put a little note here. You don't have to memorize this. You're not getting tested on near and farsightedness. Nearsightedness basically means that I can see something better when it's closer to me. Farsightedness means I can see it better when it's further from me. Okay, so nearsightedness is often described as the lens can't flatten enough to see something far away. But sometimes it's described as being where your eyeball is too long, meaning that the focal point of your image is actually in front of the fovea centralis rather than on the fovea centralis. Okay. Being farsighted, we can think of that as your lens doesn't thicken enough to focus that image on your fovea centralis of your retina. But we can also think about that as being your eyeball is too short. Okay, so your focal point is actually behind the fovea centralis. And then this is normal vision. Astigmatism is a little different because it's leading to you not necessarily getting a focal point. So sometimes it's just fuzzy in one area, and that's often due to misshapenness in either the cornea or the lens. Okay. All right. I'd like you all just to see if you can read this. So just take a minute, see if you can read this. Now, this kind of makes us think that maybe English teachers shouldn't care so much about whether we can spell. The thing about each of these words is that the first letter and the last letter are still the same. And our brain makes it make sense to us. And once we get used to that pattern, it gets easier to read. The only word I normally have students maybe sometimes struggle on is this one here, which is phenomenal. And the reason why is sometimes people don't, if you don't use that normally in your vocabulary, it's harder to pick out. So the phenomenal power of the human mind, according to a researcher at Cambridge University, it doesn't matter what order the letters in a word are. The only important thing is that the first and last letter be in the right place. The rest can be a total mess, and you can still read it without problem. This is because the human mind does not read every letter by itself, but the word as a whole. So our brain takes all of this and it makes our world make sense. So what I want to do is I just want to show you an image that I opened up here, this one. All right, so you might have seen this. Every once in a while you'll see these at a space science center, for example. So what I have is I have an image of a chalkboard, okay? And let's see if we can just make this just a bit bigger. Okay. All right, so here's a nice little image of a chalkboard. That sort of changed a little bit. Let's just go back to 100%. All right, so what we have is we've got a couple of squares marked here on this chalkboard. We've got square A and we've got square B, okay? Looking at it, because I know you all know there's a trick to this, but looking at it, which square is darker, square A or square B? Okay, so when I look at it, which square? Okay, so A looks darker, right? So A looks like our darker square. So let's do a little something here. Turn on my mouse. So what I'm going to do is I'm going to just cut a little bit out of A. There we are. And I'm going to drag that little bit of A, and I'm going to put it on B. And what we notice is it fits on B, no problem. That is exactly the same color as B. And I can do the same thing here. I can take a little bit of B, and I can cut that out, and I can move it up here to A. And A and B are exactly the same color. All right? But what our brain is doing is it's trying to make this make sense. Our brain understands patterns and it understands differences, contrast, okay? So our brain is trying to make all of this make sense to us. So what we see here is, we see that this is in a shadow, right? And I know that this is in a shadow, and therefore if it's in shadow, it needs to be darker, except I also know that I've got this pattern here. So my brain is telling me that this section is supposed to be darker, so therefore in reality this has to be lighter because it's part of my checkerboard. It's really weird. So I did include a video. I'm going to go back to our PowerPoint. We're not going to have time to watch it with this here. And it's about 3 minutes long, and it's all about how our brain uses contrast in order to detect things, understand things, and it actually enhances contrast in images. And that's part of why those little tricks or illusions work. Okay, the next thing I want to try is, I'd like you just to, if you have this, take it out. So this is my little visual looking for our blind spot, and actually I can open it up. I don't know if it would work virtually or not. I kind of doubt it, but we can give it a shot here. I think I have it open somewhere. Maybe I don't. I'll just open it this way. Okay, that should open nicely for us now. Maybe. All right. Oh, maybe not. Sorry about that. Still not going to work. Oh, there we go. Okay. Okay, I'm going to go here and share my screen right here. Okay, so how this works is you're looking at this screen, and as you're seeing this screen, you see a plus sign on the left and you see a dot on the right. Okay, so I'm going to hold this in front of me. So I have a plus sign on the left and a dot on the right. And what you're doing is you are closing your right eye. Okay, so close your right eye and stare at the dot and move the dot closer and closer to your right eye, and then the X disappears out of your periphery. And that point where the X disappears, that's your blind spot. Okay, and then you can do the same thing on this side where you cover up your left eye or close your left eye and you stare at the X and you move the X closer and closer to you, and when the dot disappears out of your peripheral vision, that's your blind spot. So usually when I do this in class, I have this on lots of different colored paper. Because what happens is really cool, because your brain is all about making your world make sense. It's not a hole. Even though the blind spot doesn't have any photoreceptors, when I stop seeing either the X or the dot, or the cross or the dot, it's not that there's a hole in the piece of paper. It's filled in with white paper. And I've tried this with blue paper and yellow paper and pink paper. Whatever paper it is, that's the color the hole gets filled up in, because your brain works to make your whole world make sense to you. Okay, so that's what's kind of cool about the blind spot. So even though we have no photoreceptors, it's not just this empty vacuous space that we have in our peripheral vision. Your brain fills it in. Okay, so that's what's sort of cool about finding our blind spot. All right, so let's go back to our PowerPoint here. Okay, so this labeling activity is just the parts of the eye. So I would recommend doing that just to make sure that you know where all the parts of the eye are. This is corrective eye surgery. It's just the laser eye surgery. So basically what it is it's using a laser to change the shape of the cornea in order to improve the vision. So the light bends properly as it's moving through the cornea, ultimately to get to the light focused on the fovea centralis. Okay, I think we are going to not look at that because I want to talk about the ear. Okay, but I would recommend watching that. Okay, parts of the ear to know. So again, we want to know the pathway to the brain. So we're going to start here with the pinna. Now, believe it or not, you learned all these parts of the ear back in elementary school. I think it's grade 3 when you do the ear. So the pinna is just the outer part of your ear. Okay, so this part right here is the pinna. So usually when we talk about ear, that's what we're thinking about, even though there's so much more to our ear than just that. Okay, so the sound waves get captured by the pinna. So the pinna is like a funnel. The sound waves get captured by it, and then the sound waves move through the auditory canal. Now the auditory canal is a little tube here, and in the sides or the side walls of the auditory canal, there is glands that produce wax. And the purpose of the wax is to capture any pathogens, okay? And then the sound makes its way over here to the tympanum. Now the tympanum is also known as the eardrum, but in our program of studies for Bio30, it's the tympanum, so that's the word we're going to use. And then, oops. So what we've just talked about, from this point here where the pinna is, all the way over here to the tympanum, that's the outer ear. Okay, and we can see we've got bone in the outer ear, right? There's a little bit of muscle. We know the ear is made up of So this is bone, that's from our skull, but we know that our ear itself, our outer ear, or pinna, is made up of cartilage. So the sound waves, they're moving, captured by the pinna, our sound waves move through, and then they bang on the tympanum. So tympanum, those of you in band have heard of a timpani, which is a type of drum. So the tympanum starts to vibrate, right? The sound waves hit it and it starts to vibrate. All right, so it vibrates, and that causes these 3 teeny tiny little bones. These are the smallest bones in your body, and they're called ossicles. Okay, os means bone. So they're the smallest bones in our body. So this first one you may have learned as the hammer. It's called the malleus. The second bone here is called the incus. You might have heard of it as being called the anvil. And this last one is called the stapes, but you might have heard of that as being called the stirrups, because it kind of looks like stirrups if you guys have done any horseback riding. Okay. All right. Then what happens? So these 3 bones here are in the middle ear. So the tympanum is vibrating. So this bone connects to this bone, connects to this bone, and they're all connected with ligaments and what have you. They're connected together, and that movement, they take this movement from this big tympanum that's moving, and they isolate that movement in this teeny tiny little membrane, which is right here where the stapes is touching, called the oval window. Now, what that does is it displaces fluid through this structure that kind of looks like a snail, and that structure is called the cochlea. So fluid moves through the cochlea, and as it moves, it's displacing some membranes, and it's actually causing little hair cells to bend. We'll get through that process in a little bit. When they bend, they depolarize, they send a message through to sensory neurons, which go through the auditory nerve. And who can tell me which part of the brain the auditory nerve sends its message to? Auditory nerve sends its message to the come on, you guys, there's 12 of you. Someone's got to go. Yeah, that's awesome. Thank you, Grayson. The temporal lobe. So the temporal lobe is getting that message. Okay, so that goes to the temporal lobe. Now there's some other stuff that's going on here that we haven't labeled yet, right? Okay, so the next thing we're going to label here is the eustachian tube. So the eustachian tube is actually a tube that goes from my middle ear down to my pharynx. So remember, pharynx is just a fancy word for throat. Okay, so from here to here, here's my draw. Oops. From here to here, actually to here, this is my middle ear. Okay, so this is the middle. All right, so that's just the middle section. And we have eustachian tube, which we had and then lost here. Let's put it back up. Auditory nerve, eustachian tube. Okay, the eustachian tube actually helps to equalize pressure on either side of the tympanum, because the inside here is the inside of my body, and then the outside is the outside of my body. So if I have a lot of pressure pushing this way, then that can actually bend and even break my tympanum. And what I understand is when we puncture it, it can be quite painful. Okay, so we can break the tympanum. So the eustachian tube, if we swallow, what that does is that now causes the pressure in the eustachian tube to be the same as outside, because it's like it opens it up to where our throat is. Okay, so when I swallow or yawn, and if you yawn, you can actually hear that yawning action, some crackling in your ears, and that's that opening, and that's allowing that pressure to equalize. So that's why when you're in a plane, when we're starting to go up, so the pressure is shifting, they tell you to drink something, so you're swallowing, chewing gum, constantly swallowing, or yawn, because that's what's equalizing that pressure on either side of the tympanum. When we have a cold, this eustachian tube fills up with fluid, and then this doesn't alleviate that pressure, and we can get a lot of pressure pushing on our tympanum, which can make it quite painful. Okay, the other part of the ear we need to know are the semicircular canals. And these are part of the structures in our inner ear that help us maintain balance. They give us a message about how we're moving, and then our body can accommodate for that to maintain our balance. So if we look at this one here, we'll call that green. So this part of my ear, from here to here, this is the inner ear. Okay, and the inner ear's got all of this bony stuff. So it's got the semicircular canals and the cochlea. Those are the parts that are in my inner ear, and we can see how there's bone all around it. Okay, all right, so those are the things we're going to learn about. So next. So the pinna, I'll look over here, let's move these around a bit. So the pinna collects sound like a funnel. So we see the sound waves coming in here. Auditory canal, so it's collected by the pinna, travel through the auditory canal, and hit the tympanum. Okay, so those are all part of my outer ear. Auditory canal carries the sound waves to the tympanum, is lined with glands that make earwax, like I said. And then the tympanum, which is the fancy word for eardrum, is the boundary between the middle and the outer and the middle ear, and it starts to vibrate because of those sound waves, so it moves. Okay, so that sound energy is causing the tympanum to vibrate. Okay, then we have these little ossicles. So in this image, the tympanum is here, this kind of purpley color, and here's our ossicles, the malleus, the incus, and the stapes. Okay, so the three bones, they amplify the sound, because the sound, this movement of this big tympanum, is now amplified. It's put into the smaller surface that is called the oval window, and that's what the stapes are hitting. Okay, so that energy of that movement is transferred to the cochlea with this membrane called the oval window sitting on it. Okay, all right. The eustachian tube connects with the pharynx. Just going to get that out of there for a second. So here what we're seeing is we're just flipping the image of what we had before. So we've got our ear now on this side. So there's our pinna, our auditory canal, our tympanum, okay? And here's the eustachian tube. It's open, so the pressures equalize between the inside and the outside of our body. Here, okay, we've got high pressure pushing on the tympanum, but there's no equalizing it because it's filled with fluid, right? And that happens often when we're sick, so there can be mucus filling it up. And that's why our hearing gets kind of muffled sometimes when we have a cold, because of that mucus buildup there, okay? So tympanum equalizes the pressure on both sides of our, the eustachian tube on both sides of the tympanum, prevents it from bulging or breaking or causing us pain. So when we have an infection in our ear, this buildup of fluid can happen, and then we don't have that equalized pressure, and we can feel earaches as a result of that. All right, the cochlea. So the cochlea, sound waves are converted into nerve impulses by a particular structure that's in our cochlea called the organ of Corti. Okay, so our cochlea has several canals in it, and we can see these canals here. Okay, and the organ of Corti is a structure that lines the inner canal. And here's a closer up look of the organ of Corti right here. Okay, and I'm just going to, so the organ of Corti has these little hair cells. These hair cells are mechanoreceptors, so they get depolarized when those hair cells bend. Okay, and depending on the movement is going to depend on the frequency of the sound waves. So different hair cells will depolarize to different frequencies, so we can hear different frequencies. Okay. All right. I just want to look at this image here so you get a sense for what's happening. All right. So we have our sound waves are coming in, and they're hitting the tympanum. So the tympanum is vibrating. The vibrations in the tympanum cause these three ossicles to move. The ossicles end up taking all that energy and banging on this oval window, which is a membrane leading into the cochlea. So what we're seeing here is the cochlea, okay? All right, so it bangs on there, and as that stapes, that last bone of the ossicles, hits the oval window, it causes this fluid to move. As that fluid moves, it causes movement in this membrane, okay? So this membrane starts to move because of the movement or displacement of that fluid. And as that moves, that causes the organ of Corti, which lines this central piece, to be displaced, okay? Now, the other thing I want to note, now it's not in the curriculum that you know the terms oval window and round window, so I will not be testing you on those terms, but it does help us to understand what's going on with this process. The round window is another membrane that's in our inner ear, okay? So the cochlea has these two membranes. One's called the oval window and one's called the round window. The stapes hits the oval window, and this is all this blue stuff here, that's fluid that fills that canal in the cochlea, okay? So it's all filled with fluid. The thing is, if the oval window, so imagine we've taken this tube and we've just extended it, okay? If we push pressure on the oval window and it's filled with fluid, there's no displacement, right? Because we can't compress a fluid. But because there's the round window at the other end, the oval window can push in, so the stapes can push it in. As it pushes in, the round window pushes out. So what we have is this kind of pushing action where the oval window pushes in and the round window pushes out. And because of the round window, this fluid can be displaced. If there was a bone there instead of the round window, there'd be no displacement of the fluid, which means the organ of Corti doesn't move, which means the hair cells don't bend. But because we have a round window, the oval window gets pushed, so the round window bulges out, and the fluid that's in the middle can displace, okay? And that allows for the organ of Corti to move. So let's just go back to the previous slide for a second. Okay, so this is now doing a cross section of the cochlea, and with the cross section of it, right, that middle canal is here, and then those other two canals, the canals on either side, that one big long canal is right here. As it gets displaced, it's causing movement here. Okay, so if I take a look at this image, it's causing movement right here. And as it's moving, right, the organ of Corti is moving up. These little hair cells that I see right here, they bang into this membrane on top of them. And as those hair cells, because they're getting moved up and down and they bang into that membrane, every time they hit that membrane, they bend. And as soon as they bend, okay, so they're sensory receptors, they send a message onto a sensory neuron, which is part of the auditory nerve. Okay. All right. So the ossicles strike the oval window. So here we see the stapes, that's the last bone of the ossicles, strikes the oval window. The fluid here in this canal is displaced, and it causes the fluid in this centerpiece where the organ of Corti is to move. And as it moves, the hair cells bend. As they bend, they depolarize. Okay. So the bent mechanoreceptors depolarize, they send an impulse onto sensory neurons, which are in the auditory nerve. So I gave you the name auditory nerve, so that's a nerve you need to know the name of. This message is processed in the temporal lobe of the brain. All right, so we're just gonna watch a little video here. Did I open this one already? Let's see, I think I did. I did not, darn it. Okay, let's go back to the PowerPoint. So guys. Okay, we'll just click on this. Okay, this should work for us.
Sound waves strike the tympanic membrane and cause it to vibrate.
Oops. Just want that to get bigger. There we go. Issues.
Sound waves strike the tympanic membrane and cause it to vibrate. Vibration of the tympanic membrane causes the three bones of the middle ear to vibrate. The footplate of the stapes vibrates in the oval window. Vibration of the footplate causes displacement of the basilar membrane.
You don't have to know the
Short
name of that membrane.
wavelengths from high-pitched sound waves cause displacement of the basilar membrane and cause it to near the oval window. This movement is detected by hair cells of the spiral organ. Long wavelengths from low-pitch sounds cause displacement of the basilar membrane far from the oval window. Again, this movement is detected by hair cells of the spiral organ.
Okay, when they use that term spiral organ, they simply mean organ of Corti. So what we have here is, go back. It's not going to do that. Okay, forget it. We're going to go to the next video. I'm just going to shut this one off. Okay, next video we have here, we'll just go back to the PowerPoint. Thought I did so good getting this all set up. This one talks a little bit more about the hair cells.
Sound waves are gathered by the pinna and directed down the auditory canal to the eardrum or tympanic membrane. Sound waves are gathered by
Just go already.
Sound waves are gathered by the pinna and directed down the auditory canal to the eardrum or tympanic membrane. The mechanical energy of the sound causes the eardrum to vibrate. This in turn causes vibration of the three middle ear bones. First the malleus, then the incus, and finally the stapes.
You just need to know them as
These tiny
ossicles.
bones amplify the vibrations. The stapes comes in contact with the inner ear at the oval window. The fluid-filled inner ear is a coiled structure called the cochlea. Vibrations conveyed to the fluid cause bending of hair cells.
I'm going to pause this. Sorry, Jaden. You're probably not the only one who can't see this, so let's just make sure that we have this shared properly. Nope, I don't. Okay, let's try that again. Just go back a little bit.
The fluid-filled inner ear is a coiled structure called the cochlea. Vibrations conveyed to the fluid cause bending of hair cells.
Okay, so what I like about this is we can see what's going on with this cross section. So our fluid runs through this part here and this part here, right? It's going to run through one part to the top and the other part to the bottom, back to where the round window is. But this structure that we're looking at here, that's the organ of Corti, okay?
If we examine this cross-section of the cochlea, the tectorial membrane can be seen at the top. In it are embedded the tips of hair cells, which are shown circled in yellow. When the hair cells bend in response to vibration, the rate of firing by the neurons at their bases changes. These signals are conveyed to the brain and perceived as sound.
All right, so remember those are going through the auditory nerve to the temporal lobe of the brain, okay? All right, next we have, okay, I will just go back to my slideshow here. Okay. All right, so that's the hearing part of the ear. We'll get to the balance part in a second. So that's, this is a labeling activity again for you to help remember the parts of the ear. This is a little video. It's about 2 minutes long. It's a bone-anchored hearing aid. My husband has a bone-anchored hearing aid. He got his first one about 13 years ago. This is a cochlear implant. So a BAHA is for people who, often for people, my husband has a condition called osteosclerosis. So his ossicles are actually, they've hardened, they've calcified, so they don't carry the sound from his tympanum to his oval window. So this method, this BAHA, allows the sound to enter a processor going through his skull, and then that message gets carried directly to the cochlea and circumvents the ossicles. The cochlear implant is for people who their sensory cells, those hair cells, don't work. So this one actually circumvents the hair cells and takes the message from outside and adds that directly to the auditory nerve. How the auditory nerve interprets that as sound is like magic. This one here, auditory transduction, it's about almost 7 minutes long. If you're gonna just watch one of these videos, watch this one. This is the absolute best video on the ear. Auditory illusion. Okay, this one we'll watch because it's very short. So let me just share this one with you. And so this one here makes us seem like the sound is always going up, and yet it isn't, because when we replay it, it sounds like it's continuing where it left off. I heard this once at a science conference session that I was at. I did some research in it, and it sounds like it's because of the octaves that are being played. If you know anything about music, playing the octaves of that sound rather than just the single note is what allows us to be tricked into thinking that it's going up. Continually going up. Okay, so back to this, and we're going to talk a little bit about balance. So, and share. Okay, so there's actually two parts of balance in our inner ear. We're only focused on one of them because the program of studies just talks about semicircular canals. So we're not going to talk about the other type of balance. But both of them have to deal with what's going on here in our semicircular canals. So those canals are arranged in 3 different planes. So we have a superior plane, up and down, a posterior, which is kind of at an angle, and then a horizontal one, which goes like this. Okay. At the end of each one of these canals, we have these little ampullas, and the ampullas have hair cells on them, and those hair cells actually bend depending on how fluid moves. So fluid moves through these canals. So one thing that I typically do with students when I'm in the classroom is I spin someone in a chair, okay? So if we're spinning this student in a chair and we're having them swivel around, what that means is that this fluid here in the horizontal canal is going to be moving, right? Now, the second we stop the student, the fluid doesn't stop, right? The student stops, the fluid keeps spinning. So what happens then is your brain is saying, hey, you're spinning based on the information it's getting from the inner ear, but based on the information it's getting from the eyes, it's like, no, you're not spinning. That confusion in that message is what gives us that dizziness. It makes us feel dizzy. So then what I do is I have a student put their head, touch their head to their shoulder, and we spin them. Now, if I'm spinning a student this direction, and then I stand them up, well, the fluid is continuing to move, but now it's moving in the superior canal, so you have this feeling like you're going this direction. And that usually causes people to become even more dizzy because we kind of know this rotational dizziness and how we feel with that. This up and down dizziness, when I was just moving rotationally, doesn't make any sense. So you usually get a lot more dizzy that way, okay? All right, so at the ends of each of these semicircular canals, you have these ampullae, okay? And there's little hair cells, and those hair cells are mechanoreceptors as well. So the fluid is moving through these canals, and as it moves through the canals, it causes those little hair cells to bend. And as they bend, they send a message, okay? Those receptors send a message to a sensory neuron, which is within what is referred to as the vestibular nerve. Now, the vestibular nerve is not curricular, so you don't have to memorize that one, okay? All right, so if we take a look at what's going on here, if we have a head in a still position, right, this ampulla, this section here with these little hair cells, well, they're not going to bend. But if I'm rotating like this, I have fluid that's moving in that horizontal canal, and that's going to make those hair cells bend. So they depolarize. They're going to send their message off through the vestibular nerve. To what part of the brain? What part of the brain is responsible for us maintaining our balance? What part of the brain is responsible for us maintaining our balance? Anyone? I'll give you a hint. It's not a lobe. What part of the brain is responsible for us maintaining our balance? Thank you, Grayson. The cerebellum. So the vestibular nerve is going to go to the cerebellum to tell us how we're moving. And then we can respond, right? So if our body's telling us we're moving too much to the right, we can respond by compensating and moving to the left so we don't fall down, okay? All right, so semicircular canals, rotational stimulus. So when you think of rotational stimulus, think of all those different movements you do on a roller coaster, because we go like this and we go side to side, right? We sometimes go at the side this way, cause fluid in the semicircular canals to move, bending and depolarizing mechanoreceptors, those little hair cells. Depolarized mechanoreceptors stimulate sensory neurons in a sensory nerve. I didn't write vestibular here because you don't have to know that, which sends a message to the cerebellum of the brain. Now, I know we're a couple minutes over, but we're going to watch just a quick 2-minute video on balance. Okay, so I already have that open, so we'll just share that with you here.
The vestibular system is a sensory system responsible for providing our brain with information about motion, head position, and spatial orientation. It is also involved with motor functions that allow us to keep our balance, stabilize our head and body during movement, and maintain posture. The main components of the vestibular system are found in the inner ear in a system of compartments called the vestibular labyrinth, which is continuous with the cochlea. The vestibular labyrinth contains the semicircular canals, which are 3 tubes that are each situated in a plane in which the head can rotate. Each of the canals can detect one of the following head movements: nodding up and down, shaking side to side, or tilting left and right. The semicircular canals are filled with a fluid called endolymph. When the head is rotated, it causes the movement of endolymph through the canal that corresponds to the plane of movement. The endolymph flows into an expansion of the canal called the ampulla, within which there are hair cells, the sensory receptors of the vestibular system. At the top of each hair cell is a collection of small hairs called stereocilia. The movement of the endolymph causes movement of these stereocilia, which leads to the release of neurotransmitters to send information about the plane of movement to the brain. The vestibular system uses 2 other organs, known as the otolith organs, to detect forward and backward movements and gravitational forces.
Okay, so this is a different system. So this is what allows me to know basically just my head rather than my body movement, okay? So this is the one we don't need to know.
There are 2 otolith organs in the vestibular labyrinth: the utricle, which detects movements in the horizontal plane, and the saccule, which detects movements in the vertical plane. Within the utricle and saccule, hair cells detect movement when crystals of calcium carbonate called otoconia shift in response to it, leading to movement in the layers below the otoconia and displacement of hair cells.
All right, that is it for his video. Let's just make sure that this is going to play. Go back here. And there. Okay, so what I would do right when you're done with me here is I would just watch that video that we looked at. Just going to go backwards for a second here, that we did not look at, that you should look at. That's about 7 minutes long, auditory transduction. So that's this one here, just because I think this one is probably the absolute best one as far as sound. So just take an opportunity to watch that. And oh, now I have some more videos trying to play. And yeah, so that's it for today. So the worksheet that I gave you today, that is due Monday, and then you'll have a quiz on special
So Lit Mobile just sent me this solar wireless battery pack. I'm excited. Let's see what's inside. I really like the build. It's got hand grips on the side, it's shockproof and water resistant.
I knew that was going to happen. All right, we'll just not share anything here. What's that? All right. Okay, so are there any questions about what's happening, what's coming up, anything like that? All right. So make sure you do that worksheet, watch the video that I asked you, and again, email me anytime you have any questions or anything. Monday's class, because we did do the notes on special senses, so I think Monday what we'll do is we'll meet, probably not for 45 minutes, maybe more like 20 minutes, and you guys can just ask some questions that you have. And then we'll move into reproduction on Wednesday. All right, any questions? All right, everyone, enjoy yourselves. Have a wonderful weekend, and we will see you briefly on Monday, so it won't be a full lecture on Monday. Okay, talk to you guys later. Bye-bye.